Okay, well, thanks very much to the organizers for inviting me to speak. I'm going to complement, I think, a lot of the things that you just heard, because in academics, we don't have as much money to do those pivotal phase 3 trials that you were just hearing about. So first, I thought that I would try to define what translational research was. So first, clinical research is research that is patient-oriented, where there's direct interaction with human subjects. So that's a little bit different from human subjects research, whereby you'll be working with non-identifiable tissue specimens and that sort of thing. So that's considered human subjects, but not clinical, where you're actually dealing with patients. Translational, at least the T1, the Translational Block 1, according to the Institute of Medicine's Clinical Research Roundtable, is sort of what you always thought it was, bench to bedside, where you transfer a new understanding of disease mechanisms gained in the laboratory into the development of new methods for diagnosis, therapy, and prevention, and their first testing in humans. Then there's a second type of translation, which, according to the article I read for this, said is really kind of misleading because it's not related to the first type of translation. But I would tend to disagree with that for reasons that Dr. Stevens just mentioned, and that's that we have to think about taking things and having them realistically implemented. So the second type of translation is where you take those results from clinical studies into everyday clinical practice and health decision making, where you do things like outcomes, things like informatics come into play here, global health, public health related issues, where you implement discoveries. So I think that that should be considered, and that those are the two types of translation. The bench to bedside, though, gets a lot more money, frankly, about 10 times more money in the United States than the T2 block type of translation, which actually one could think of as being even more impactful, because frankly, public health related projects can more easily in some ways save lives than the bench to bedside stuff that we tend to focus on here. So translational research is very important. The United States has these CTSA centers at various universities, and there are 60 now. There's about a $500 million annual budget as of 2012. These are facilities that facilitate translational research in an academic setting. They provide oversight with research coordinators and whatnot. Not so much, I'm learning, boots on the ground. In other words, they don't tend to have people that will shepherd patients through trials, et cetera, but they kind of provide oversight. Also in Europe, there's a big investment. There are a couple of journals, various foundations. Industry is getting into translation also. So it's a very important aspect of our research. So what are the steps? So this is, I never had any training in translational research specifically, so it's been a little bit catch as catch can for me, like I think it has been for pretty much everyone in academics that's doing this. But these are some of the steps that I just, as I was making the talk, thought about that we tend to go through. And this does apply primarily to radiopharmaceuticals, which is sort of my area of interest, but we'll talk about some other agents as we go along. So number one, probably the most important thing, and this was alluded to in the previous talk, that your preclinical data has to be impeccable before you go to the trouble of translating something. And I have seen things that have been translated in the literature, human studies, that if you took a look at the preclinical data that was also published, you could tell that this is not going to work, that the pharmacokinetics are not there, the signal to noise is not there, for imaging. Nevertheless, because of the momentum of going to the trouble of trying to make a nice agent and knowing it could be first in class and all that kind of thing, people take it through all the way to the clinic without that compelling preclinical data. So that's point of advice number one. You want to avoid that. Then you have to consider where your involvement ends. So when I was in grad school, I remember I was a chemist, and all I cared about was making the compound, making the radiolabeled compound, which we did at WashU I was at Illinois, actually, but we traveled down to St. Louis where they had a cyclotron. And if I made the compound and handed it off to the biologist, I was very happy because I'd spend a week optimizing the radiosynthesis, and I felt like I was done. But then I noticed that we'd give these to animals, and if it actually worked and hit the target, then I kind of got interested in the biology. And then I said, gee, that's great. But then after it works in a few rodents, you want to put it into a non-human primate, see if it works in that. And then you see it works there. You go, well, anyone could do non-human primates. What about patients? So then you get it into patients. And then after that, you think, okay, so what? I did it in a patient, big deal. Is this going to become the clinical standard, right? So you have to sort of in your mind decide, where you're going to log off of this. And for better or worse, in academics, we're sort of forced to log off somewhat early. But in terms of commercialization, and this again was emphasized very nicely in the previous talk, if you're not going to make money off of this, if you can't convince investors that they're going to make money, and imaging is tough to make money, you are not going to be able to commercialize or get to those pivotal phase 3 trials where you really see how this thing works in the real world in multiple different patient populations. And to do that, you've got to get approval by the Center for Medicare and Medicaid Services. And there are extremely few PET tracers that have been approved by CMS, but the reason that it's important to get approval for them, that means that Medicare and Medicaid will reimburse for those studies, that they have a definitive indication that go on what are called ICD-9 codes in radiology departments. They could bill for that, and you can't really do that for very many molecular imaging agents, unfortunately, at this point. Okay, so where do we get off? Think about that. So then you take a look, so translation devices, so investigational device exemptions versus drugs, which is investigational new drug applications. In our business, it's really more along the lines of drug, but there are, or IND, but there are plenty of molecular imaging scientists that have been working on devices. For radiopharmaceuticals, we're fortunate that we could use an exploratory IND mechanism, or we could even use the local radioactive drug research committee, and I'll talk a little bit about that. An EIND is basically when you can give less than 100 micrograms to the patient. It's what they call a phase 0 study just for pharmacokinetics, just so you could see whether this thing is going to be worth pursuing and doing it in a human being, which of course is the most relevant possible model. So that's great when you can do that sort of thing. You can't do it with drugs. You can't really do it with even MR-based agents or, frankly, even optical agents, and we can talk about that too. But radiopharmaceuticals, you're going to be there, and I'll talk about why in a little bit of detail. And in fact, to be honest, and I should probably ask, is there anybody from the FDA here? Okay, well, then I could say that I have found that they sort of have been treating our EINDs like INDs. So EINDs also, you have fewer patients, maybe 30 patients can be so, but we, have multiple indications, more than 30 patients, and, we send in our reports, and I haven't heard any complaints yet. So EIND and IND seems a little bit blurry right now, at least for radiotracers. So toxicity and dosimetry, very important. Dosimetry, of course, for radiopharmaceuticals is next. Then you have to send things to your local, IRB, Institutional Review Board, that should say. And we have specialty boards, and then we have the overall university board, specialty meaning if you're developing an oncologic tracer. But it's really the first in human study, I think, that we in academics try to focus on, or should frankly focus on, because phase 2 and 3 are very difficult unless you're doing strictly clinical research and are funded to do that, usually in conjunction with a company. So this is why radiotracers seem to be able to be translated more easily than other probes, and that's the tracer principle. So if you give 5 millicuries of 5,000 curies per millimole, and I apologize for the Europeans in the audience, but I'm still not used to becquerels, but in any event, if you give this to a 70 kilogram person, you're giving 5.5 times 10 to the minus 6 mgs per kg of a mass dose. And when you give that, you get an image like this. This is the thalamus. That's where the mu opioid receptors are. and that is a tracer dose. On the other hand, if you take this exact compound, carfentanil, and you give 2.5 mg per kg, that's what they use on the Discovery Channel to take down elephants. Okay, that would be a lethal dose in a human being. So we could give compounds that are lethal at tracer doses and give them, and that's why we can translate these things. So the toxicity package, as was discussed, is definitely not as elaborate as it is for drugs. So I'm going to show 4 examples of things that I have experience in translating go from difficult to easy, and I'll explain why, and it'll be very clear why some things are harder than others, but nothing is really that easy, frankly. But so prostate cancer, to start, this is our favorite target in our group. We look at prostate-specific membrane antigen. We've been studying this since the late 1990s. But step number 1, good target and compelling preclinical data. So this target, we like it because it's on the cell surface, it's associated with aggressive disease, it could be general for other cancers, it does have some biological significance, it's a marker of androgen signaling. So good target. This is our composition of matter, the compound that we have synthesized, or one compound to try to bind to this target. So you know that's another good thing to consider. And then I guess maybe if you could click on that you have to have a spinning mouse. And I'm sorry, if you could click on that thing on the left, that mouse picture, yeah. So this is a dynamic PET scan that Jürgen Seidel provided for me a long time ago. But it shows there's 2 tumors in the animal, one underneath each arm. And you can see these are isogenic tumors, so they're exactly the same, except one of them expresses the target, PSMA. So we inject this, we could see that we're only going to the tumor that expresses target. So this is pretty good preclinical data that something right is happening, and we can generate time activity curves in a rodent model. So then, as I mentioned, you have composition of matter. If you're ever going to get this thing translated for real, and let's face it, all of us are in this business because we want to see our compound become the clinical standard that is used worldwide, at least for a couple of years, until it gets displaced by something else. I mean, that should be, your goal. That's our goal. So we don't pursue anything unless we think that it's really going to go forward. And unfortunately, you need to have protection like this to do it, because then companies will be interested in licensing the patent, and they're the ones, frankly, that are capable of taking it further on. Okay, so once you have your composition of matter, you've got your nice preclinical data, you're thinking, I might want to take this forward, you apply for your patent, then you need toxicity. And back in the 2007, they had what was called the DECIDE program at the NCI. Now they have the NEXT program. And these are programs that do enable toxicity studies. They will pay for toxicity. They will also pay for the synthesis of precursor that needs to be synthesized according to good manufacturing practice, which I'll talk about in a minute. But the point here is that there are mechanisms. Back in 2007, it wasn't so bad. It was like come one, come all. We're looking for applications. Okay, 2013, these are, at least for me, a lot more challenging to get. And we've had a couple of applications that I thought were pretty good that were not funded. So it's getting a little bit more competitive. So before you do the tox study, though, you have to do a CGMP synthesis. This is synthesis according to good manufacturing practice. This already is pushing it. Okay, so at an academic center, we used to do these ourselves, but the FDA has made certain aspects of this extremely onerous for an academic center. Now, to do a GMP synthesis, the synthesis that we published, it was not a GMP synthesis. It was a very straightforward synthesis, but now our synthesis has to be something that we can automate and give to patients. And what makes things GMP is, not the brilliance of the protecting group strategy, but it's the fact that every single thing you do has to be very carefully documented. You have to have the scales that you use to measure out your compounds with have to be certified. The person certifying the scale has to be trained and certified to certify the scale. It's just like too much. Okay, so that's But we've done it for some of these earlier compounds. It takes about 6 months to do this, and Ron Meese is the one that worked that out. Then you have to automate your synthesis, and we do that. This is definitely not an advertisement, but this is just an example of an automated synthesis box. And the way we make this compound, there's actually 2 steps. The first step is shown here. We make this fluorobenzyl bromide, and to make that compound, this was the synthesis that we use at Johns Hopkins was adapted from the literature from this publication, and you can see that that's going to take some time. So it's not trivial to do that. If you're just trying to do discovery work, you don't need to do any of this. In fact, you can do everything by hand, but you can't do that anymore for GMP. Then you have to be able to formulate this for injection. And the formulation even is automated so that we get to the very last step where we sterile filter it, and then voila, you give it to the nuclear medicine physician and they inject it. Okay, so you can see that this is a fair amount of work. So you don't want to do this if you don't have good preclinical data, right? Okay, then once you've done the GMP, you give the material. Usually you've got to make about 1 gram of this stuff so that they can do rodent toxicity studies. And as was mentioned, we only have to do generally 1 dose, mimicking what we would do for an imaging study, and then we do just one time dose, one time dose, and we do acute and subacute toxicity studies in a single species rat. And frankly, I think it's great. They just let you do rats, and then if that works out, it's not toxic. You have to give amount of compound that goes up to about maybe 100 or 1,000 times more than what you think is going to be an adverse effect level in humans. And if that's not toxic, then you send that data to the FDA. Then you have to fill out your IND application. You'll notice, by the way, that this application is 102 pages long. So it's, again, not for the faint of heart. Probably the hardest part on one of these applications is the chemistry, manufacturing, and control data. And we do that in close collaboration with our PET Center, who are experts at doing this. So they develop a certificate of analysis. They're the ones that we work with closely for the automation. And that has to be very detailed. We send that in to the FDA. They've got 30 days to get back to us. And then usually on day 29, they have questions. And then we have to answer their questions. And then eventually, you get this very happy letter that says study may proceed. Okay, so then you can go ahead and get started. And then, this, again, there are a lot of people involved. Steve Cho is the guy who actually, does the injections and moves these into the clinic for us. He's the PI on the IND, on the project, and the author of the IND. And Tiffany does the interfaces with the FDA. And then eventually, voila, after many years of effort, you get a human study, and it better be good, after all that effort. So, this literally took close to 10 years since the first time we made the compound in animals. So, you got to ask yourself if you want to do all that. So that was a reasonably, that's a standard PET agent, single injection, high specific activity. So what if you want to make something a little more complicated? So a lot of people are making dual modality agents, right? There's a big emphasis on that. So we basically took one of these PSMA agents, Sangeeta Ray, and we made it into both an optical as well as nuclear imaging probe. Now, why would you want to do that? And that's because on day 1, you could inject the patient and do a SPECT scan. And then if the target is pretty much located only within the prostate, of course, this is an animal, and anyway, this just shows that we're hitting the target in a standard xenograph. If it's localized to the prostate, on day 2, you could actually do optical imaging with the exact same agent without having to give another injection, because optical imaging is actually very sensitive. Okay, you can't microdose at this point in time. At least we can't, and conversations I've had with the FDA, patients generally need about 10 milligrams of this stuff if you're going to do it. for intraoperative guidance, which is why you would do this. So this is more for staging. If the staging looks like it's in the prostate, then they're going to go for surgery. You could use this for enhancing surgery to make sure there's no positive margin. So dual modality probe, a little bit more complicated to translate. Okay, you got a couple of different things there. So then you go to the Army and you write to them and you say, I'd like to try to translate some of these things. And this is the usual answer I get, not recommended for funding. And I could tell you that that's exactly where this compound is going to stay until I have enough energy to write another grant, or until somebody picks up the patent, etc. But that's sort of where it's going to stay for now, because that was a fairly expensive program. Okay, so the other thing you could do, now you've got a multifunctional theranostic nanoplexes. So this is a project in collaboration with Zavir Bujwala. And you can see this sort of Rube Goldberg device where we can deliver siRNA to choline kinase, bacterial cytosine deaminase. We can label this with probes that are sensitive for MR, for PET or SPECT. And we actually do get targeting in vivo. And actually, I'm not going to show the data, but this does work to turn off choline kinase, and it does work to have an activated or an active bacterial cytosine deaminase. It activates 5-fluorocytosine to 5-fluorouracil. The key behind nanomedicine is just to get things to happen where you want them to happen at the tumor. Okay, so that's why we do this. This, I think, would be an extremely valuable thing to be able to translate. But we've got siRNA, we've got optical, or actually this one doesn't, but we have had optical agents on here. We've got a big enzyme on here. It could be immunogenic, genotoxicity, immunotoxicity. I mean, there's just so much. The toxicity package for this would not be the $55,000 or $60,000 that might be for a PET agent, but it might be more on the order of $300,000, and you have to go into dogs and who knows what else, maybe chimpanzees or, I don't know, small children or something. I don't know what I should say. But it's not easy to get something like that. Now, this is really hard, nanoparticles. I mean, how many of these have been in patients? Not that many. You could probably count on one hand, maybe two. So this was one, again, our target, PSMA. So a company called Bind Biosciences, they licensed a patent that we had for putting PSMA on the surface of drug-loaded PLGA nanoparticles. So this is something that I would love to have done in animal models and then in patients. Absolutely hopeless. We had to have this. We just got lucky that they picked this up and decided to translate it. So then they, I don't know what they did, but it was definitely not cheap to move this nanoparticle. I should probably know what kind of animals they used. But they did eventually get this into patients, and they had a nice result where they do show that they can use these particles to deliver docetaxel at doses that are lower than are normally given systemically, because you're concentrating it all where the cancer is. So a very compelling thing to do, too expensive for an academic lab. So this had to be licensed out and done by industry. Okay, so these will go more quickly. So infection is another thing that we've been looking at. This is a project in collaboration with Bert Fogelstein at Johns Hopkins. So Bert is interested in treating cancer by giving bacteria that are anaerobic and will home to that well-known anoxic core, or I should say the hypoxic core of tumors. The idea is you give these anaerobes, and the anaerobes get in there, they home to that hypoxic core, they proliferate, they take out the tumor, bacteriolytic cancer therapy. Okay, so Bert asked us a long time ago now if we could image that process because it was going to be hard to get the FDA and the IRB to approve giving patients bacteria to treat their cancer. Okay, you could imagine that would be challenging. So to prove that we have this under control, we wanted to be able to prove that we just had the stuff in the tumor. So you could see here, this is a tumor, and we gave this Clostridium novii, which is the bacteriolate. This is given systemically as spores. It gets into the tumor, munches on the tumor, actually produces gas in here, and basically that's sort of what's happening. if we give radiolabeled FIAU, we were very lucky, and we noticed that the FIAU would be able to be phosphorylated by the bacterial thymidine kinase, just like it is by the transgenes HSV thymidine kinase that are put in for molecular genetic imaging. So that was a fortuitous discovery, but here we can prove that we've got the bacteria is only in the tumor, it's not in the brain, it's not in the chest. We could show this to our IRB and they say, okay, go ahead, you could do human studies with that now. Okay, so we were able to do that, but we have to be able to give the FIAU. Okay, so where are we going to get that? Oh, the other thing is more compelling clinical data. We could show that the FIAU is specific, and we could even calculate how much bacteria there is using standard curves. We could try to get the number of bacteria just based on the images. Okay, so we're trying to do sort of what we saw before with the amyloid agent to prove that this thing is binding things. Now, the problem with FIAU, which is also known as pheoluridine, is that this stuff is lethal. So it caused, this by the way would be the imaging agent. This caused 5 deaths back in the early 90s, very unfortunate. But because we work at the tracer level, we should still be able to give this to patients. And in fact, when we published our first papers where we use I-124 for PET, which by the way, I-124 is about $1,000 a millicurie. So it's very hard to make. You have to let the I-123 decay away. So each one of these patients that we did cost on the order of $10,000 to do. That price is going to have to come down if we're going to be able to use this stuff broadly, or things like it. But anyway, I wrote a letter to the editor here because there was an article where they were talking about ways of imaging infection, and they said, well, this stuff is not going to work because it's going to be hepatotoxic. Okay, and they say it's hepatotoxic based on this study. So, and those were nuclear medicine physicians that wrote that. So I wrote in and I specifically said that we are giving 9 times 10 to the minus 7th milligrams per kilogram, too low to have a pharmacologic effect. So the reason I show this is it shows that you got to just keep fighting, okay, to try to move these things. So even though they know about the tracer principle, they see that there were deaths, hepatotoxic. This is never going to be hepatotoxic. It's just always too low of a dose. Nevertheless, you have to write to, this was our Radioactive Drug Research Committee, so we didn't get an IND for this. This was in the public domain. This has been used by Andreas Jacobs and Yuri Galavani have used these things in patients. So I referred to their articles. I got every single clinical study that ever used FIAU that proved lethal. And you could see that these guys are getting 1.5 or 1.2 grams of this stuff at 1.7 mg per kg per day. And that's, when you get the lethality. So RDRC is something that you can use when something in the literature has already been given, only in 8 patients, but I was miraculously able to convince our IRB to do it, and we did show that we can image infection with this. These are orthopedic infections. It's particularly good for orthopedics because of the pharmacokinetics of FIAU. And that was another example of translation and some of the steps that we had to go through. So real quick, inflammation is another thing that we've been interested in. There's a lot of things in the brain in particular that involve inflammation, like traumatic brain injury, even Alzheimer's disease, schizophrenia, psychiatric disorders. So Michael Cassiu, who was a long time ago a fellow at Johns Hopkins, he's now at the University of Sydney, he found a compound in the literature. So you could find some compounds you have to make from scratch, some you find in the literature. So I was at a meeting with Michael a number of years ago now, probably around 2005, and he had a compound called DPA-713 that Michelle James had tested. This is your compelling preclinical data. This is a baboon. This shows the PET agent. This shows the PET agent with a compound of another class that binds to the same target, which is a translocator protein, nothing in the brain. And this shows the central benzodiazepine receptor ligand that does not block the agent. So this also used to be known as the peripheral benzodiazepine receptor. So this is the best data you can get, pretty much. A non-human primate, you show complete receptor blockade, Okay, very compelling. So the NIMH has a program also called the Toxicological Evaluation of Novel Ligands Program, and you can get radiotracers in there too, although that's becoming quite competitive. They paid for the toxicity studies, which, were a couple 100,000 in this case. And we did do human studies based on that toxicity. For this one, we had to write an IND or an EIND. But now we've got this very nice tracer that we're using in multiple different types of brain inflammation. So it can be quite worthwhile to do that. And last, I want to talk for a minute about, I say metabolism, but really it's more sort of working with other modalities like MRI. Okay, what can you do with that? Okay, these, you'll notice these are under the easy category. All right, so I hope I'm not offending any MR researchers here, but they seem a little bit easier than the PET, and I think you'll see why that it is indeed easier. So this is a paper recently published that we recently published that Xing Yang, who actually is going to talk about more of this kind of stuff later on in this meeting, using things like salicylic acid as an MR contrast agent, using chemical exchange saturation transfer or CEST imaging. Okay, so I won't go into the details, but the bottom line is, if you're going to start using stuff like salicylic acid, which is, like aspirin-like, which is acetyl salicylic acid, then you're getting close to stuff that we've been giving to people for hundreds of years. Okay, so that might be a little bit easier to translate. Of course, the key is the dose. So, what kind of, for MR, it's not that sensitive, so we probably have to give a supratherapeutic doses. But nevertheless, it's not like giving a completely bizarre new chemical entity that requires more complex toxicology. To do even better than that, so Peter Van Zyl at our institution, he doesn't like to do MR where you actually got to give something to the patient beneath human skin. Okay, he likes to develop pulse sequences that will pick things up, again, using CEST, in this case with glucose. So you infuse glucose, you have to infuse it, it's at a fairly high concentration, and eventually you can actually see changes in the brain. And you could imagine that you could do this with brain tumors, for example, etc. So using just glucose. So this was very easy. I asked Peter how long this took him to translate to humans, which he has done. Okay, I'm working with him on this, and he does have a clinical protocol. I basically just asked our research coordinator to, talk to the IRB about it. She put in a protocol, and we gave the amounts of glucose. I mean, patients get glucose every day, and next thing you know, we're inpatient.
Due to technical difficulties, this is the end of this recording.